Hello, everyone. Good morning. My name is Nicolas Gaudenzio. I'm research director at INSERM at the Institute Infinity in Toulouse, France. And I'm also chief scientific officer of the biotech Genoskin, located in France and in the Boston area in the US. Today, I'm going to share with you our last publication in Allergy, in which we define a new way actually to understand immunogenicity of a molecule that would be injected in the human skin at the tissue level, at the single cell level. And for that, we use natural human skin that we preserve alive outside of the body. So we have developed one of the best way actually so far to maintain human skin alive for more than 7 days. And we can perform sub-Q injection into this human skin. From one donor, we can perform multiple models, and then we can compare dose. We can do a longitudinal analysis over time, etc. What is very interesting with this technology is that we maintain a full immunocompetency of natural skin resident immune cells. Then, as a proof of concept, we use the Moderna vaccine to understand the immunogenicity that would be induced within the skin tissue as compared to water for injection. What are the biological process? What are the immune cells that are targeted, etc. We combine different methods such as proteomic, bulk, and single cell RNA-seq analysis, mRNA vaccine tracking sequences, and also multiplex annotated tissue imaging system. And using this data, and I encourage you to download the paper, what we define is that at the tissue level, there is not only the immune system that is activated, but there is a complete perturbation of the human skin immune ecosystem, starting from keratinocyte, dermal blood vessels, lymphatic vessels, and also the skin immune cells, obviously, that are modified in a certain way after the injection of the vaccine. And we track at the single cell level which immune cell would incorporate the vaccine and what are the consequences on those immune cells of the incorporation of the vaccine in terms of antigen presentation, mRNA processing, secretion of proteins, etc. Here is just a small glimpse into our research article. And I think this is one of the most interesting parts is after the injection of the COVID vaccine, we sorted CD45 positive immune cells and we perform a single cell RNA-seq analysis at 8 hours and 24 hours within the same donors. And what we understood is that when we track the spike sequence from the Moderna vaccine is that it's whatever the route of administration, sub-Q intradermal, for example, it always goes within the dermal dendritic cells and macrophage compartments. And for 60% of it, it goes into mast cells. And we found that very interesting. And then we can perform a perturbation analysis to see what are the genes that are modified after the incorporation of these vaccines. And we could define exactly what are the sequential events that happen into the cell types after the incorporation of the Moderna vaccine. Then another very interesting piece of data, it's related to Mantis, so our multiplex annotated tissue imaging system, which enable in 3D to analyze distribution of proteins in immune cells. We have defined a myeloid panel to understand antigen presentations. These are some biomarkers that we're using. So you can draw the full immune landscape and the digital map actually of the tissue and understand what are the biomarkers that are upregulated. And when we did that, we were really fascinated to see that the injection of the Moderna vaccine in the human skin actually triggered the aggregation of antigen-presenting cells near blood vessels and lymphatics, and those cells are highly enriched in maturation marker of co-stimulation, antigen presentation, and here you can see on the right side, actually in green, of CCR7, which is one of the key molecule involved in chemotaxis to draining lymph nodes. And with that, I hope I have encouraged you to download the paper. And thank you very much for your attention. And if you have any question, feel free to reach at me. Thank you.